Good morning. My name is Ronald Vos. I represent SensID, where I run the business development. And today's symposium is about controls and reference materials, and we gave it the title The Good, the Bad, the Costly. The good and the bad is pretty obvious. There are good and there are bad controls out there, but what defines a good control? The costly is an interesting one. We are often being confronted with the fact that people think that controls are expensive or costly. What we would like to do is give you a little bit of background on how controls are made, what kind of QC measures are in place, and then let you decide whether they are costly, expensive, yes, no. The choices you have when it comes to controls are pretty obvious. There are basically three choices. You can decide not to use controls at all. You can decide to design and develop your own controls, what we call homebrew, and you can actually source commercially available controls. We take a look at the first, not use controls at all, and every now and then we encounter customers that don't use controls because they believe they have enough positive and negative cases to justify that their instrumentation is correct. However, if you look at the need for controls and you're going to look some more in literature, you find these overwhelming amount of EQA schemes where a lot are focused now on clinical genomics, and then in clinical genomics a lot on circulating tumor DNA. Here is a collaboration of 4 different scheme providers, indeed, IQ, NPAF, ASBL, and they run a study for the analysis of reporting circulating tumor DNA. The scheme error rate was 20%. 20. 1 out of 5 did not report correctly. Another one from the same group where there's very interesting consensus actually that the sensitivity of the method should be stated, and you're basically looking at the total amount of cfDNA because it will vary. It's recommended that you do quantitation. But if you do quantitation, how can you do quantitation without providing or using a control that is exactly quantified to establish indeed the ground line of where you're working on? One from the same paper, which is an excellent paper by the way, perfect read for everybody, is the issue that the absence of proof is not the proof of absence. It's a beautiful sentence, but it means so much. The fact that you don't detect a variant or mutation does not necessarily mean that it's not there. It is just a matter that your current technology, method of choice, does not detect a mutation. You only know if it's not there if you use a control where it's certified that the mutation is not there, and the other way around. Add the mutation, run the detection. If you find it's correct, if you not find it, you have an issue in your protocol, in your technology. So it's a very important one to remember, especially when ctDNA goes lower and lower and lower in requested AFs. Another paper, Pre-Analytical Issues in Liquid Biopsy from 2020, Michael Fleischhacker and Bernd Schmidt, and they speak about the increasing analytical sensitivity. We at SensID, as no others, know about this because we just started a collaboration with Saga Diagnostics from Sweden. Fantastic young organization building very, very sensitive ctDNA assays, and we will work with them to provide controls that go beyond the 0.1% allelic frequency because they have the ability to detect it. So there the choice of the controls are becoming very important indeed because the cutoff values are at a certain level. So the 3 choices, not use controls at all, is basically a no-go. Just use controls. You have to. You decide on the treatment for a patient. You decide on what the status of a disease is when recurrent, yes, no. So without control, how can you do that? Then the 2 other choices, design and develop your own controls or source commercially available controls. We want to focus a little bit on how we look at controls, how we make them, how we do the QC on controls. And to tell you more about that, let's go a little bit in the background of the organization. So we are a German organization based in Rostock. We are pretty young still, but growing massively. We are ISO 13485 certified, and we develop control reference materials for clinical genomics, and we apply a number of technologies for this. The products that we make are under the highest standards, developed and inspected under QC conditions, and especially about QC conditions. We're going to talk a little bit more about that. We make bespoke or contrived materials, and we are a supplier of these kind of materials. And often these materials are compared to homebrew, and they are viewed as equal in quality. I doubt that. there will be homemade materials out there which are really, really good, but there will plenty out there which are not as good. Let's dive into the world of controls. But first, something about contrived materials. I'm from the Netherlands, as you undoubtedly can hear from my accent, but if you look up contrived and you translate that via the different machines that you have and even dictionaries, you find that contrived means gekunsteld. Well, gekunsteld is actually strange because it's viewed as fake, not real, unnatural, so it has a negative tone to it. Well, if I bring my car to the repair shop and I get it back like this is really gekunsteld. But what we want to show you that there is nothing gekunsteld about our controls. We make them as commutable as they can, but we keep in mind that they are designed to be used as a controls and very often in a quantitative manner. So with the coming explanation, we want to show you why source controls, like companies from like SensID, are very often outperform homemade controls. Where and when are controls needed? Let's start there. Well, we live in the world of evolving biomarkers. If you thought a couple of years ago we had the EGFR gene, if you now see it in a random ctDNA panel for EGFR, how many ctDNA variants are to be found in EGFR? It's incredible. So evolving biomarkers lead to the question of a certain technology development or selection of technologies to actually follow, detect, analyze these biomarkers. The testing of that technology can then be done by using a control, because using clinical samples would be very cumbersome. You first need to do perfect quantitation, for instance, of these samples, and how much of the sample do you have? A control is there basically like an endless supply. After the testing of the chosen technology, you will do your in-house validation. You run it over multiple operators, multiple times a day, different circumstances and so on. And from that moment on, you go to clinical and analytical validation. This is where real samples come into play and are really needed. But still, for all this trajectory here, you can use a control material, contrived material, for instance. Then you're going to start in routine use, and especially in routine use, where you quickly could see deviations from baseline, these kind of things. Another aspect where controls could be functioning really, really well. Now, the way that we look at developing a control is that there are many, many questions to be answered. So if we look at contrived material, or let's put it like this, where we make a control with synthetic fragments, and in this case a cfDNA 6-plex, we start with contrived material, variants that we buy. We have to think of the background. What kind of genomic DNA do we put in the background? Because cfDNA exists against the background. So, and that gives us, with the ratios, the allelic frequency. So we need to really determine what kind of background we're going to use in the case of this 6-plex cfDNA. Then what variants are we going to choose? That's typically known. But then comes the question, are they flexible? Can you actually bring them together in one material, and that one material can indeed be analyzed by the technology of choice, and you are able to differentiate one variant from the other? The AF per variant typically, because not all variants might have the same LOD, for instance. And then the fragment size. There's a lot of discussion on fragment size, and Björn will go in his next presentation somewhat deeper into the aspects of fragment size for variants. And then very important question, what sample matrix? If you look into the literature, actually controls, especially for ctDNA testing, should be based on plasma, because you need a full process control. You also need to look at extraction efficiency. But buffer could work, then you miss a step in your certification, let's say, your validation, because you miss your extraction, but still it's doable. Now let's go to what would be the line of thinking when you do approaches with cell lines. Well, with cell lines, you would need to do a cell line selection. So you look for the variants in a specific cell line. If you're lucky, you find a cell line with 2 variants instead of just 1. And then you need to look at the specific AF in that cell line. You need to do full characterization, because it could well be that in that cell line there are other variants, actionable mutations, that in time might need to, they might trigger a reaction from you, and you might not want them in there or want them in there. So you need to do characterization of the cell line. You need to determine the AF, the allelic frequency, before but also after mixing. If you mix different quantities of genomic DNA with variants in the background at a specific AF, you change the total amount of genomic DNA in the background, and your allelic frequency will change likewise. Can you still reach the required AF possible? And then after all that work, you're actually gonna detect it, you're gonna run an assay, and you're gonna adapt the mix to make sure that it actually fits to your needs. Then you have a final product for QC. So quite a long way to go. Several questions still to be answered though. The questions to be answered are for which technology and which process. We have found on cases where a specific control does not work for a specific technology. Because of either fragment length or double-stranded, single-stranded DNA. A lot of factors play a role in this. So you really need to know, if you develop a control, what technology is going to be used. What concentration DNA per microliter is given here, because it could well be that you need to dilute massively because the concentration of the control is way too high. If you dilute massively, you introduce error, and you don't want error because you want to do quantitation, as we have seen from the EQA scheme. Quantity and shelf life. How much are you going to make? How much of the material will you make, and how long will it last you? Typically, if you make a product, you can rely on literature for a certain shelf life, but after that, you will need to do shelf life testing. So you can make 10 microgram of something, but if you're not going to use that in a period of 5 years, the product will expire and you have nothing basically that you can do with the product. Very important aspect, but it more relates to SensID than any other company in this field, is whether the control is to be used for RUO or IVD or in-kit control. We have, thanks to proprietary material that we have available, the ability to develop an IVD certified control. So if you are thinking of implementing a control in your kit, in your assay, we can actually do that for you. And we come with a CIVD certified control, add it in the kit, and your work is basically done there because we have done that work for you. Could be a lot of work taken out of your hands if you go that route. You move into the design, the development and manufacturing phases, you end up with a product, but then it starts. Then you enter into QC. And QC in an organization like ours is massive. There are more people working on QC than anything else. So for QC on controls by SensID, the tests and technologies that we apply are for DNA quality and concentration, the bioanalyzers, the qubit and UV-Vis. And in this, and Björn will talk a little bit about this as well, we focus on single-stranded and double-stranded DNA because both are very important in establishing the quality of the control and the proper use of the control. Then you have digital PCR assays for the quantitation of the final product and the intermediate product. So in that process, we need to select and acquire specific digital PCR assays, but we need to validate all of them. We just can't use them off the shelf. We really need to validate these assays. And next, because we're making a multiplex, remember you're making a 6-plex here, we need to validate the cross-reference. Do they not have influence on one another? So it's a lot of work to be done in the QC of a final product. What we do in the final product QC is that we follow strictly, very strictly, ISO 2859-1. And in the example where we, for instance, make 100 vials, you actually go to a table that you can find in this ISO method, and you find between 91 and 150 vials. You need 8 vials for full QC. So for any lot of controls that we make that is more than 91 vials, we fully QC'd 8 individual vials from the final product pool. Now what does it mean? What does it mean as in work? What are we actually doing? Well, for this example, 6-plex C of DNA, we run for assay validation alone. We run gradient digital PCR in triplicates at 5 different temperatures, totaling up to 300 wells. For specificity digital PCR, we run them in triplicates again, up to 42 wells. Then we move on to product validation. So this is just assay validation, there's product validation. Digital PCR in triplicates on the test pool, again 36 wells will be used. And then digital PCR in triplicates on the product pool, and again 36 wells. So you see what amount of work goes into a control before it's available for you as a commercial off-the-shelf material. So which one now to use, the homebrew or the commercial one? Well, just weigh the facts. Commercially available, we have a product that is developed and manufactured under ISO 13485. I can't say anything either negative or positive about the way that homebrew is developed and manufactured. I don't know what ISO standard has been followed, if any. I only can comment on what we do. And we follow strictly this ISO norm. We follow strictly this ISO norm for the QC procedures. I don't know what's being done in the homebrew environment. I hope they do it like this. I just don't know. Shelf life testing is done extensively on our site, because if we make a product that can last for 3 years, that would benefit indeed us, because development costs are pretty high. If shelf life testing is done in the homebrew situation, probably, I don't know again. Here, however, something that we do know. The product that we design is made platform agnostic as much as possible, so that it can function on any kind of technology. What we typically see in homebrew applications is that it's designed for a platform that is in use at the moment, but the minute that you add another platform to your portfolio of technologies, then suddenly it could be that the control doesn't fit. Here, what is need to be done by you is purchasing only. All the other work is done. And here the product is tailor-made. You indeed have made a product that is perfectly fitting your need. Perfect. I mean, that's really the way it is. Here you just need to talk to us, which could be okay, could be interesting, and we fine-tune indeed your needs, pricing and all these kind of things, but it's a fixed product in the end. What, however, is important, especially when we speak of multiplex, think of the example of our 6-plex, we don't use plasmids. We just developed a product for a specific group of people, a DPYD genotyping control, by recommendation from a professor from the Erasmus University. And he asked us, tell me again, you haven't used plasmids, have you? Because I don't allow plasmids in my lab. No, we're not using plasmids. And very often in homebrew, plasmids are used, especially when we come to multiplexing. Coming to the negatives there, the product is expensive, but I put it between brackets because it's perceived as expensive, and here the product has a low cost, but again in brackets because it's perceived low cost. The product has a fixed configuration, totally true. This, however, is often made via plasmids, and a lot of labs should try to stay away from plasmids. If you get ever contamination, then your whole lab is contaminated. And what is negative, of course, about a homebrew is that many hours a day have been used. We speak very often to customers that indeed spend so much time on developing a control in the end to come to a conclusion that it doesn't work. We do this every day. It's our job to make controls. Commercially available, the product is expensive. Let's take another look at that objection. This is, for instance, an off-the-shelf ctDNA product that we have available, that we just made. We just guided you through the process of the work that needs to go in there. You buy one vial, 400 nanogram of DNA. It's around 150 euro. 150 euro, one vial. But it contains 400 nanogram of DNA. Now this is ctDNA, so your typical sample input would be anywhere from 10 to 20 nanogram if you would mimic real-life samples. That would give you 20 to 40 runs. 20 to 40 runs is €3.75 to €7.50 per run, but for 6 variants. So you cover 6 variants in one run for that amount of money. Commercially available, the product has a fixed configuration. True. Here's an example of a SensID 8-plex EGFR control that we sell a lot. It's the 8 very common, very often required variants in there, but it's fixed. We can't change that. However, customization of a control that you need is always possible. We can always make you a control. I'm honest about it. It will not be the price that you just saw. The price will definitely increase, maybe very high, but it all depends on the plex. The higher, the more costly. The quantity that you need, to a lesser extent, because the material itself is not so expensive. But the timelines, if we can make you a control that could last 3 years, you can run 3 years from the same batch, and all this time you don't need to buy new material. That's worth something, because you also don't need to change controls in the time of the process. And there you have usefulness of the product. The usefulness of the product is, maybe the product is so good that we can actually think, hey, this is commercially interesting to bring this to the larger audience that we serve, and we take the development cost on us. So what I would like to say with this last slide is actually, if you can't find what you're looking for on our website, talk to us. We run many schemes, EQA scheme providers, pharma and so on. We have many products that are not on our website, or we can potentially make for you, where pricing could be maybe very high, but maybe not, because it could well be that, like with the DPYD genotyping product we made, we think this is an interesting product. The market needs such a product. We'll take the development cost, and all our customers need to do is just pay for the off-the-shelf material. That will end my presentation. I would now like to ask Björn to take over from here and give you a little bit more insight in the deeper details of a control made especially for liquid biopsy. Thank you very much.